Hi, um, Jamie, come up.
Hello.
Everything good? Did you sleep at all?
I got a couple of hours, and I've also figured out the Google Drive thing. I've got it sorted now.
Cool. What? It was so late yesterday. Hi,
Yeah,
everyone. We opened the room a little bit earlier since Dr. Sergey Krasnokutski is new to Clubhouse. So we opened a little bit earlier to give him like a chance to get used to the app for a few minutes beforehand. So yeah, stay tuned. We'll start in around 8 minutes. So, thank you. Will be really exciting. Hi, Serena. How are you?
Hello.
hello, Serena.
Good. Hi, Jamie.
Pretty fun talk today.
Oh,
Aliens and stuff in space causing life. I think that's awesome.
yeah, will be really interesting. I agree. Let me add the topics. If you see, oh no, he should be right here at the speaker panel.
You know, Katerina, I have to thank you and your club because the stuff I've been learning the past month has mean that when I've been in other rooms, I've actually had knowledge to share now that I've learned from here.
Yeah,
Absolutely. Like, do you know that the topic came up of like AIs making art? I remember we had that casual room a couple of weeks
yep,
back. Yeah, and I was like, I know these things. And I'm starting to understand what certain biological things mean as well. So I'm kind of like feeling smarter all the time. Thank you for that.
yeah. Somebody also actually said that his dinner conversations he can turn it way more interesting lately because he knows so much more things, like different things. So
Yeah,
It is
I,
funny. So I'm starting to use our rooms as examples in conversation.
we do, yeah, totally. Um,
yeah, so it makes us more attractive because we are smart at dinner conversation.
what gets me is, as I heard somebody in a different
So,
room
yeah,
talking about.
go ahead.
Oh, just and somebody in a different room, and they were talking about some like consciousness, and they were talking about other stuff. And given what I've been learning in the last month, I was like, yeah, that's not how the brain works. But you carry on. Because it's one of the. Have you spoken to people in which you would actually think, if I didn't know what I knew now, I would probably be nodding my head and agreeing with what they're saying. But given the stuff I've learned, you kind of go, yeah, I can see why connect those dots, but that's just they got that from a picture book, not from reality.
Yeah, exactly. Yeah, welcome everyone. We'll start in around 5 minutes. Thank you for coming. And yeah, it will be a really exciting talk. Um, really looking forward to it. So I hope everyone is having a good morning. What time is it for you, Serena?
It's coming up on 10 in the morning.
Oh, okay, yeah, it's the same
Did you have
here.
your coffee, Serena?
The first cup is half empty.
Oh no, Serena, it's half full.
Well, when it's your first cup of coffee, half empty is better.
Right, okay. I see what you're saying. Uh,
Yeah.
have you having have you having a are you having another like night on tonight of the casual science society tonight, or are you not going to bother because of
Oh
the room
yeah.
today?
Should we do it? I was thinking about doing a casual room. Are you guys up for it or
I mean, I'm up for it.
okay? Yeah. Okay.
I'll see how the evening goes, but if I find myself like I often do, checking Clubhouse for what's going on. Sounds good. I stayed up way too late last night, but it was in a room where somebody asked me if I buy into the panspermia idea. Oh, is that the aliens from space? But because one take on this is if if we are if we can show that peptides can grow in space, it just means all the more that they can grow anywhere given the
I would,
conditions are right. And so it still doesn't imply that that's where life came from or that it seeded life. It still could have happened here just as feasibly, but it just shows it's ubiquitous and it's a stronger statement that there's probably life all over the place. We just got to find it.
I had that absolute thought when I was reading the paper. Actually, that exact thought. I was thinking, like, what does that mean? Like, shouldn't there be more life now if it's all just floating out there? But we don't really understand enough about the sheer probability numbers, right? And considering the fact that we can't really travel beyond our own solar system, even so, for all we know, there could be teeming life all over the place. We just haven't, found it and
It,
waved to it yet.
it does because there's a lot of arguments pin on that, oh, it was so improbable that, there must be something else going on. And studies like this just chip away. No, it's actually quite probable at that stage. And no, that step's probable too. It
Yeah,
just chips away at the probabilities.
it's funny that this paper actually gives credence to, like, well, you know how there was a crazy idea that we originated from space things, but a lot of them got really crazy, like aliens literally dropped off some humans and everything in between that. But this is something that actually sounds probable. Like it makes a kind of sense, that there's so much space and so much stuff out there, and that life, how is it Jeff Goldblum says, life finds a way.
Hi Frank, how are you?
Oh, did I mute? I think I muted myself. Sorry.
Hi, Katerina.
How's
I'm
how was
doing good, and this uh, research very interesting. I read the paper a little bit, but not very in depth. But it's definitely very interesting. It bears on the question of origin of life, right? So the macromolecules. It's very interesting. So yeah, at a very low temperature in space, and a very plausible route for yeah.
Definitely. That's what we were just saying there. It sounds so plausible. How you doing, Frank?
Hi Robert, how are you?
Hey, I'm very well, thank you. I think, yeah, I'm all right. Just listening to it reminds me of is it Russell's paradox? Like where the hell are they? But you're right. I mean, the space and the vastness of what we seem to perceive out there is just mind bending. Is it? I think the known edge of the universe is something like 45.5 billion light years away or something. It's ludicrous. So there's plenty of space. But I also have a sense that if anything has, I just want to just explore this one thing briefly. The Entangled Life book, which I'd highly recommend, speaks of fungi existing and surviving and even thriving in space. So that I found that quite interesting. It wasn't really taken anywhere as far as I remember by Sheldrake in the book, but I thought about it quite a lot since, and I was like, well, if those dudes are quite happy sort of hanging out in space, then presumably there's an outside possibility that they could have travelled here. The same as there's an outside possibility, a reasonable possibility that peptides could have travelled here, or like you say, they turned up here and went the other direction. I think the vast to the small is probably a more more defensible argument. Not that we know, of course, but I certainly don't know. But the idea that they've come from the vastness of space to this tiny little planet on their way through, I think is probably, yeah. Anyway, I'll think about that some more. But thank you.
Yeah. You had the idea that it's not more like human stuff was floating around in space till it all crashed on Earth, but more like Earth had like all of the ingredients bar one to like bake a cake, which is called a human, and the stuff from space that was just the last right amount of flour or sugar landed just right.
Yes.
Like so it's less like
What?
humans came from space.
But you gotta if you're gonna go there, you gotta say that that last bit of stuff couldn't have possibly happened on Earth. It couldn't have been all over the place anyway, and it just added to the pile. And what's cool about papers like this is once it's shown that it's thermodynamically stable to form, well, then it flips the whole argument. It's like, why would you think that it wasn't already here? It's
Mm,
stable. It forms. It's natural. It's there. It's everywhere. It'll just happen. It
you
even
mean you?
happens in space. But
Oh yeah. Um, do you mean like do you mean like it was all the Earth already had all the ingredients? You mean?
well, so once it's if you're going to hold that thermodynamics works everywhere the same, it's a
Hmm.
universal, then you no longer have a reason not to, or I mean, the burden becomes on you to say that no, it couldn't have happened on Earth. But there's
Ah,
something,
I can
so
tell.
it's just a natural process. Peptides will just form spontaneously under the right conditions.
I think it would
Right.
be. I think it would be fair to say, thinking about it a little bit more, that potentially it was both. It's not and or.
That's my
Because
point. Exactly. It's
we
everywhere.
think,
Yeah.
yeah, well, we think of the
Hmm.
Earth as a closed system, which is tempting to do because largely it is, but it's not true. There's lots of things come and go, and and particularly coming inbound. Um, and are you familiar with? I've never really checked this out scientifically, but are you familiar with the idea that, and again, not a certainty, just a hypothesis, that water wasn't originally earthbound. That came in from externally. Um,
Well, water just forms. I mean, it's hydrogen oxide. It just forms.
yeah, I'm not entirely sure that stands up entirely.
Oh, sure, it does. I mean, chemistry is my background. Yes, it does.
That actually
Yeah,
does
no, I
make
no,
sense.
I,
But
I, I understand that, but then it may be that those conditions for that to happen did not exist on Earth prior to something coming in.
So,
Well, the frequency of some of those molecules, well, in particular hydrogen and oxygen, is very. I mean, they're well, hydrogen, of course, is the most abundant um,
Element.
uh, element. Yeah, exactly. Sorry, I'm just waking up here. um, and then oxygen is actually pretty abundant as well. So from kind of early stage fusion events of stars, there's quite a lot of oxygen. So um,
so yeah,
I think it's
oxygen
reasonable
is
to coalesce around one planet. Basically, that's the reason
it
I'm saying.
pervades the crust and from weathering in rocks, and hydrogen's quite abundant. Yeah, there was where water can form, and and we're in that Goldilocks zone where liquid water form. You'll definitely get it, and it's there's no reason for water to have had to come from post-Earth formation meteorite deposition.
So how did we get from an entirely molten ball to water that boils at 100 degrees? How do we make that transition?
I think how was it the atmosphere was actually made? Because didn't that start to protect us from like rays and things like that a lot more? Didn't that protect the planet from um certain kind of external stuff like solar winds and stuff? Because wasn't Denny's talking about that? I don't
I
know how
don't,
the atmosphere actually formed though.
So there's several mechanisms to form water because water is thermodynamically stable. So there's the precise mechanism. I haven't spent too much time on that question because it hasn't really posed a problem to me. mean, stable molecules will find a way to form because they, as you know, they're thermodynamically downhill, and the free energy is
Mm-hmm.
favorable for them to form. So there can be multiple mechanisms to arrive at stable molecules.
Actually, so I guess
Right.
Robert does bring up a good point. I'm just actually thinking about this because let's imagine, yeah, you have a molten planet. How would you have? You'd either have to have some type of hydrides, like things just possessing uh, hydrogen bound to it, or you would need obviously oxides, and then somehow those things then reacting to form water. So I'm wondering how, yeah, would you not maybe start coalescing other things like he's saying, getting pelted by comets or otherwise,
Well, just
after cooling basically of molten material.
the heat within molten material itself is sufficient for the activation energy. In essence, whatever form of hydrogen you're in, the oxides would oxidize it and you get water.
Yeah, so I guess I'm just thinking, like, what is the actual composition of the molten material then that would allow that to occur? So that's my question.
So the composition of the Earth, oxygen is in all kinds of rocks, and just weathering processes. What I'm curious about, we had a reducing atmosphere earlier on, and so there's certainly a lot of abundance of nitrogen and amines and ammonia. So it's quite possible that the hydrogen in the form of ammonia oxidized, and water would form there. But there's other sources of hydrogen, whatever form that hydrogen is in. If you've got molten oxides around, you're going to get plenty of water.
It's a fascinating idea that like you're talking about the simple chemical combinations to make things like water and stuff like that. It's actually kind of fairly basic, right? That the simple clashing of stuff together in space can form that. It's actually not surprising, and that actually can start to be shaping planets. I mean, I can't help but think that we keep losing track of the kind of timescales that we always talk about. You know, we were so used to mentioning things like billion years. It's like that's a really, really long time,
Well,
you
there's
know,
some
for
theories that that the moon formed in a big impact event, and it's quite, some of the scenarios that have been worked out were quite fascinating in that. But if that's the case, the composition of the primordial Earth is well represented by the moon. Where all the water event went, it doesn't have the gravity or the atmosphere to hold it. But
mm,
we've recently found water underneath the regolith on the moon.
really,
Yeah, it's in minor but um, mineable concentrations, and so there's initiatives to investigate how to extract water out of the regolith of the moon. But that, in a sense, is a snapshot of the composition of the primordial Earth cooled down. And yeah, we remember when we used to think that there wasn't much water on Mars.
mm,
Yeah, now there's actually quite a bit. Um,
I didn't know that. I've only ever heard like sci-fi books say when it keeps saying there was water on Mars, and there's not water on Mars, and there's water on Mars, and there's not water on Mars. So there's
it's
actually water on Mars.
Oh yeah, well, the polar ice caps are huge deposits of water,
Cool.
but there's also water underneath the surface that it can be held there, is if it not exposed to the atmosphere. Just dig a little bit down and you'll find it.
Here's my next weird question for you, Serena. If there's water on the moon, that's cool, but why bother extracting it? I mean, we don't need it. Is it
Well,
just to make a new brand of Coca-Cola?
we don't want to have to transport it. Talk about your expensive beer.
We
Oh,
are
I
we
mean, it's
are
so.
we are at heart we are extractive primates. So that's
They
what
would
we do.
they would so make beer out of
Moon
water from the moon.
beer.
They would,
What will the
yes.
workman's, the international economy, what will the workman's beer cost on the moon?
Well, it's like, everyone says,
Uh,
"Oh, my water is kind of the best. It makes the best beer. It makes the best, whatever, whiskey or whatever."
yeah.
And we only source our local water here to make the
Right.
beer. Right?
Local lunar water.
It makes you lunar.
Yeah. I can see the ads flicking through my head right now. It's practically selling itself already.
Who's going to open the first bar on the moon?
Um,
Actually, it probably, yeah. Hey.
Hey, Dr. Sergey Krasnokutski. Ha, there he is. Hi, welcome. Thank you so much for
Hello.
coming. The unmute button is all the way on the bottom on the right hand.
Hello, yeah.
Hi,
Hi,
Welcome,
welcome.
you made it.
Thank you
Doctor.
Hello.
for
Welcome.
coming. Welcome to
Yeah,
Science Society.
yeah, thank you for the invitation. So, I would present this recent publication about the finding of peptides.
Yep. Let
Okay,
me introduce you really quick to the audience so they know
okay.
that they know you a little bit. Yep. Welcome everyone to the Science Society, and special thanks to Dr. Sergey Krasnokutski
Yeah,
for coming today. And just to give you a little bit of background information, he obtained his PhD on the low temperature study of the molecular structure of biomolecules and the Institute for Low Temperature Physics and Engineering at the National Academy of Sciences of Ukraine and the Max Planck Institute for Flow Research, Göttingen, Germany, in 2004. And then he did his postdoctoral research at the University of Kentucky, Lexington, USA. And since 2008, he's a member of the Laboratory Astrophysics Group of the Max Planck Institute for Astronomy. And yeah, as I said, his research focuses on low temperature molecular physics and chemistry with a special interest in structure dynamics and formation pathways of biologically and astrophysically important molecules, as well as their complexes and clusters. Thank you so much. We are very honored to have you here today. Thank you.
yeah, thank you for introducing me. Okay, yeah, I think I will now give the presentation without showing any pictures, but I saw that the article is shared, right? So that everybody in principle can have a look there. But I will start with a short introduction why we are interested in this study. Maybe you know the idea of panspermia, that the life was brought to Earth from space. There are different types of this idea. It could be that life is brought already as living organisms which are brought from different planets. So asteroids could crash into other planets and take some part of the material from this planet and bring it to space, where it can fly for quite a long time and finally arrive on other planets. And then it can cause a life on those planets. But this, of course, doesn't explain the origin of life, the first origin of life. And there is another molecular panspermia idea, which says that some organic or biologically important molecules can be formed at low temperature areas of space. Then these molecules can be brought on the board of meteorites or comets to the planets, and there they can cause the origin of life. They could either facilitate or even be responsible for the formation of life. So, and in our research, we wanted to test exactly the latest idea and to see how far the chemistry which lead to the origin of life can already happen in the low temperature areas of interstellar medium. There are many actually evidence for this later molecular panspermia hypothesis. For example, many organic molecules and biologically important molecules such as nuclear bases, amino acids, sugars were found in meteorites. But of course, comets material is expected to have even much wider variety of biologically important molecules. And our idea was also to test how these molecules can be formed. So in space, they are believed to form on the surface of cold dust particles, which are flying in space. The dust particles consist of refractory core, which is usually either silicate or carbonaceous material, and this core is covered by molecular ice mantle, which consists of water, ammonia, CO, CO2 molecules in most. And there are different species from the gas phase which accrete on such dust grains, and their reaction on the surface cause the formation of new molecules. And of course, some molecules can be formed also in the gas phase directly, but their formation is limited because the associative reactions are not allowed. The associative reactions are only allowed when there is a presence of the third body. The third body is anything basically. It can be atom, molecules, or it can be the surface of the dust grains. But in the gas phase, the collision between three molecules is very unlikely, and that's why such reaction is usually believed to take place when there is a surface of the dust grains where the species can accrete, and then this big surface can work as a third body. So, first we studied the investigation of formation of glycine. For this, we study a reaction of carbon atoms with ammonia molecules, and we got quite interesting results there. First of all, we found that in spite of the presence of energy wealth in this reaction pathway, the proton from nitrogen can be transferred to the carbon atom, and it happens at any temperature. Basically, we managed to study this reaction at 0.37 Kelvin. And the other interesting finding in this reaction was that the product of this reaction was in the high-line triplet state, so it stores quite high amount of energy. So this reaction energy can be then used to trigger subsequent reaction. So we might have a chain reaction. Okay. Can you hear me?
Yes.
Okay, because I just got an email about the issue with the app from you. But okay, let's continue. So then we also managed to see that subsequent reaction with water and CO molecules may lead to the formation of glycine, but there was quite big energy barrier which prevent this reaction and make it less probable. But then we also thought about the next step, whether we can polymerize glycine at low temperature, and what we found out that it was not possible at low temperature of the interstellar medium because there was quite high barrier for this reaction, and it consists of detaching of water molecules. Basically, when two amino acids meet together, one of the amino acid gives OH group and the other amino acid give up hydrogen atoms. This species combined and water molecules go away. And in this way, peptide bond can be formed. But of course, the detaching of OH and H requires a break of chemical bonds, which has quite high energy barrier for the processing. So on the next step, we also found that the formation of glycine also has high barrier for the attaching of water molecules. So, and then there was idea, of course, to skip the step of attaching and disattaching water molecules. And we thought about the molecules, which is called aminoketene. Unfortunately, now I cannot show you the images, but if you open the article, you can probably see this molecule structure, and it was an isomer of monomeric component of glycine peptide. So if you open the article, you could see the figure 2, and there the aminoketene structure can be seen. These molecules can be polymerized much more easily compared to the lysine molecule, because here you don't need to break any chemical bond. You just need to transfer the hydrogen atom from nitrogen to carbon atom. And we wanted to study the formation of this aminoketene molecule and tested it experimentally. Unfortunately, till now, this molecule has never been detected experimentally, although there were many theoretical predictions that this molecule should be stable. So to do this, we used experimental setup where we have silicon substrate, which is cooled down to very low temperature, let's say 10K. It is placed in ultra-high vacuum chamber, which is mimic the pressure of the molecular clouds in the interstellar medium. And then we deposit on the substrate our reactants, basically CO molecules, ammonia molecules, and carbon atoms. And when we deposited these molecules on the substrate, we can see the formation of aminoketene. So the first result was already quite exciting. But the next result was even more interesting, because when we started slowly rise the temperature of the substrate, we could see that infrared spectra of the deposit on this substrate changed. We can see the appearance of peptide bond, which is a characteristic bands of peptide bond. And from this result, we can conclude that aminoketene molecules polymerized. But this was not expected theoretically. That's why we need further confirmation of this result. So when the temperature of the substrate increased, we took it out from the vacuum chamber and performed ex situ analysis. We solvated all deposit on the substrate in the water methanol mixture and provided to orbit-trap mass spectrometry analysis. This is a very high resolution mass spectrometry analysis which can provide unique information about the elemental composition of the molecules. Here we were able to detect many different molecules which are formed, but the important finding was that most of these molecules were arranged in the series, and in this series separation between the mass peaks exactly matched to the mass of aminoketene molecule. So we knew that all what we observe on the substrate is a polymer of aminoketene. And for some of this series, we were able to predict the molecular structure, which was modified peptides. I say modified because what we observed is not a classical peptides. It doesn't have NH2COOH terminals like classical peptides. Instead, it has NH2 terminal on one side, but the other terminal is a bit different. It might have CH2 groups or NH2 groups, but we didn't see any COOH group. And the molecular structure of these peptides was also confirmed by the collision-induced dissociation. Basically, those ions of peptides were sent with a high energy to the cell which was filled with argon atoms. And by collision, they fragment. And how they fragment tells us about the molecular structure of initial ions. And this fragmentation pattern match to the predicted molecular structure of the peptides. In this way, we knew that these peptides which we observed in our experiment was formed at low temperature, and it was formed at the conditions which is very similar to the conditions which are present in the interstellar medium. So, and in our experiment, we use the reactants which are the most abundant in space. These are simple carbon atoms. Basically, most of the carbons in the SM consist in the form of atomic gas, up to 80%. Then ammonia molecules and CO molecules, which are also one of the most abundant molecules which are present in the interstellar medium. Therefore, we can suggest that this chemical pathway may actually cause quite considerable abundance of peptides which are formed in the molecular ice mantle covering the dust grains. And just to explain later, all these dust grains would aggregate, forming all solid materials which we observe in our solar system. Of course, during the planet formation, all this would heat up, and all organic will be destroyed. But later, this organic can be brought to the planets on the board of comets or asteroids, where the pristine molecular composition of the interstellar dust can be stored. And of course, the delivery of such peptides to the planets raises the question about the possibility that they can trigger the origin of life. Actually, right now, the most common theory for the origin of life is RNA world, where RNA molecule was self-catalyzing, so it was autocatalyst, which trigger the development and the formation of other more complex species. But in a view of new findings, we can also suggest that probably proteins or peptides contribute largely to this chemistry and also lead to the formation of more complex molecules. And finally, all this chemistry brings us to the origin of life. So, thank you for your attention. So I would be happy now to answer your questions.
Yeah,
Thank you very much for that. That was fascinating.
please go ahead, everyone, with questions. Flash your microphones, and yeah, thank you.
So thank you. Wonderful talk. I've taken a fancy to this topic, particularly the kinds of chemistry that would lead up to the RNA world. Looking at your mechanism, going through the ketene intermediate, so the first question I have is, I mean, this seems like a very general mechanism into polymerization of these peptides. Could you comment on the perhaps the use of clay, the phyllosilicate clays, as the catalyst third body in this polymerization reaction?
No, in fact, we didn't use any silicate clay. We didn't use specially any catalyst. We performed this experiment on two different kinds of substrates. One, it was pure silicon substrate, and the other substrate was potassium bromide substrate. On both substrates, we got absolutely equal results. So in this case, we could exclude that substrate act as a catalyst or give any influence on the chemical reaction. But from the other side, it's true that our quantum chemical calculation showed that two aminoketene molecules have some energy barrier to polymerize. And therefore, of course, question arrives, why did they polymerize at low temperature? And the most probable explanation is the presence of ammonia. And ammonia is known to act as a catalyst. For example, I don't know whether you know, but the first detected polymer in space is POM. That's a polymer of H2CO molecules. And in the lab, they found that these molecules can polymerize at low temperature in case if even small quantities of ammonia are present. So ammonia are known to be quite efficient catalyst, and yes, I believe that in our case, actually, it was ammonia, which is also present in the molecular ice mantles on the dust particles, worked as a catalyst.
Well, that's fascinating that ammonia itself would. And is that the NH3 or is the ammonium ion?
No, it's ammonia NH3. Yes.
Uh-huh. Okay. So it's fascinating to think of if ammonia alone will catalyze the possibilities of how ubiquitous this chemistry could be under such a variety of conditions. It sort of paints a picture that peptides can form um, could form in outer space, of course, as you've shown, but also on the conditions of the primordial Earth as well.
Yes, that's right. And also, I would like to highlight another aspect that the new finding is really in the fact that peptides may be formed by the polymerization of aminoketene molecules, and the aminoketene molecules has definitely much lower barrier for the polymerization compared to the amino acids. For example, if you would start with a glycine, which is amino acid, you would not be able to catalyze their polymerization with ammonia, so because the barrier there is simply too high. But when you start with aminoketene molecules, yeah, the polymerization happens much easier.
I had a question. So, thanks for a great talk. It was really, really interesting. How, so you kind of demonstrated a relatively simple peptide-like polymerization product. To what degree do you think, so, you mentioned kind of the RNA world, and people have suggested that there are alternatives to that, kind of an RNA peptide world, for example, where you don't need the complexity of something like the ribosome to make peptides, along with potentially RNA molecules that could then help both co-evolve each other, basically. And so, in the case of these peptides that are formed, to what degree could you have peptides with slightly longer, say, side chains still undergo some of this surface chemistry? Is there a limit to the complexity of, say, substrates that could participate to make, say, a longer alpha amino acid side chain?
Of course, yeah. In our experiment, we can see the polymerization of aminoketene, so it means only glycine peptides are formed. But yeah, what I learned from literature that some photochemical reaction with replacement of the side chains is quite common. So basically, the proton can be replaced by any other species that would cause the formation of different amino acids. Moreover, if you look on the molecular structure of aminoketene, you may find that on the carbon, alpha carbon, it has only one hydrogen. So basically, there is nothing which is attached at this position. So when it polymerizes, we assume that the proton from nitrogen goes to this carbon, and then it becomes a glycine amino acid. But of course, if you would have some other reactants present, not that clean condition as was in our experiment, we might expect that this alpha carbon may take not only hydrogen atoms, it may also attach some other chemicals, and then another amino acids would be formed. But this experiment, of course, are only planned, and we don't know yet whether it will work in this way or not.
Thanks.
But I'd add that it's certainly known chemistry that you can have substituents in that position and for the ketene intermediate to form. So it's quite plausible to consider this mechanism as a route into other peptides, right?
Yes, I think the mechanism which allows the side chain replacement is quite well known. I didn't study such reaction by myself, but I read few research articles where it was described in details, and it looks like quite straightforward way which would allow the formation of more complex peptides. And of course, we will probably try it soon.
It'd be fascinating. I could certainly see it as an organic chemistry homework problem, given the result for lysine. Show how you could do the rest of the peptides that are known.
Yeah, that would be quite interesting for sure.
Thank you very much for the talk. This is a slightly more general question, but the things that you're discovering right now are absolutely fascinating, and I won't lie, they're a little bit above my head. But is this indicating that there's a much larger chance for life of some kind to have developed in other places then than we currently thought?
Can you please repeat?
With what you're discovering that so much of this can happen in space, does this seem more probable that life could be forming on other planets and, stellar bodies than what we thought?
Yes, I don't know what is currently assumed for the probability of the formation of life on other planets. I think it's even before our finding, it was considered as very probable. But yes, with this finding, that the chemistry can be so simple and lead to the formation of peptides already at this low temperature, and the peptides can be delivered to any exoplanet, then yes, I would expect that it should increase the probability that we find life on other places beside Earth.
Quite exciting thought. Anyone, any more questions? Does anyone have any more?
Can I speak?
Go for it. Yeah.
Yeah,
Go
thank you
ahead.
very much for doing this room. I would like to congratulate Dr. Krasnokutski. Did I get your name right?
Yes, thank you.
Yeah, congratulations on the paper. This is all very fascinating. Our former collaborators, when I was in the US, they have been working in the area of prebiotic protein evolution. And my mentor's main focus was on the ribosomes. So the thing is, there are two ways of looking at the origins of life, to put it in a broader perspective. One is a top-down approach. If you look at extant life and look at the universality and conservation levels, all roads lead into the core of the ribosome, the protein-making machine, particularly the PTC, peptidyl transferase center. And that is one of the many reasons why the RNA world idea by Walter Gilbert got a lot of credence. However, there are problems with the RNA world, and because still now there has been no demonstrated evidence of an RNA replicase. But having said that, the recent emerging ideas include cooperative mutualism between peptides and RNA, in which both cooperate to stabilize each other. So the kind of work that you're doing and your results are absolutely fascinating in this context. Um, we know very little about the prebiotic world because it's like a shot in the dark, right? We go all the way down to the ribosome, the PTC, but then what happened before that is an absolutely dark area. And the kind of results that you have produced are one of the many lines of evidence to show that peptides could have been formed in multiple ways. And eventually, as the ribosome developed, the genetic code was evolved, and the stage was set, and we now have the extant ribosome as a universal molecular machine. So I just wanted to add those points, and if anybody is interested, I've actually posted a couple of papers in the chat section. One paper talks about the mutually stabilizing interactions between protopeptides and RNA, which was published in Nature by Luke Lehman and Moran Pinter. There is in around 2020, and then there is another paper which is just now being it's on the it's a preprint that's available. It talks about how thioesters provide a robust path to prebiotic peptides. Now, these are all multiple pathways, and the work that your paper has presented is quite significant in this context because there could have been multiple pathways in which peptides were formed in the prebiotic world, but eventually, when the protoribosome started making peptides in a rather shabby manner, not an efficient manner, and slowly evolved, the genetic code was established, and then the rest, as they say, is history. I just wanted to add those comments. But again, thank you very much for doing this room, and congratulations again on your wonderful paper.
Yeah, thank you, and thank you for the information. Yeah, it's indeed sounds quite interesting, and yes, I believe that in principle the origin of life doesn't go through RNA only. I would expect that proteins or peptides should also contribute on the early stage, and especially with this finding that probably peptides were available from the beginning on our Earth, they for sure somehow contribute to the chemistry.
Absolutely. Even the ribosome itself is a ribonucleoprotein complex. Even though the PTC itself, its function is mostly mediated by the hypothetical proton wire transfer between in the PTC region, which is now being described as being comprised of two entities, two symmetrical entities by Ada Yonath's group. Ada Yonath, she won the Nobel Prize for the ribosome structure in 2009. Her group has been doing some phenomenal work over the last several decades, and recent evidence suggests that, in fact, there's a very recent papers that came out in the last month where her group has broken down the PTC into two parts, and they have variants of the PTC, and some of those variants can also produce a peptide bond. So the structure is more important than the sequence itself. Now, as the ribosome developed, of course, it needed proteins to stabilize it. So there was definitely cooperativity between RNA and peptides. And of course, once the genetic code was established, I'm repeating myself, the ribosome completely took over to synthesize proteins. Yeah, but again, thank you. I would be more than happy to back channel those papers if you're interested, Dr. Serge.
Yes, that would be of course interesting. I think I read some of your articles, but also in this context, I would also like to mention the other thing that peptides and proteins are actually catalyzed by itself. For example, in human body, they performed most of the catalytic activity, more actually than RNA. And in this sense, their participation on early stage on the chemistry, at least to my understanding, could be important. But I just start to doing chemistry on this large molecule, so I probably don't have complete information.
Well, it's interesting to ponder how the ribosome came to be, and what this paper, what your work shows is that peptides, we can more or less take them for granted as being present. They have spontaneous formation routes on fairly generic surfaces, and we separately know that the purines and pyrimidines of nucleosides can form from hydrogen cyanide condensations. Those are classical experiments. So we can assume both the purines, pyrimidines, as well as peptides uh, have routes to be fairly common. And a problem in more complex chemistry is escaping the entropy and reining that in and bringing structures together and catalyzing them so further more complex reactions can form. And part of the fascination I've had with clays is that they both can provide substrate um, amazingly compatible geometries for arranging structures in well, I can get to that, but also managing ions and in the form of wet dry cycles, condensing phosphates and in formation of nucleotides, condensing those. But also the peptide chemistry is less known, and what this result does is chip away at the starting materials for those condensation reactions can be comfortably assumed to be present, and the monomers and of peptides are quite likely around. Uh, but the geometries in the clays, if taken through wet dry cycles, can provide the scaffolds and the surface chemistry to catalyze these condensation reactions. And in an environment such as riverbeds, where you have continual wet dry cycles from diurnal fluctuation, it can it's a very plausible, so it seems to me anyway, means of condensing larger peptides and condensing larger nucleotide structures, but also providing the scaffoldings to bring these structures together so they can interact. So just some thoughts, but it's a fascinating topic, and I think this is important work on the peptide
Can
side.
I add something to what Dr. Sarina just said?
Sure.
Yeah,
Yeah.
yeah. Thank you very much for bringing that up. The current ongoing hypothesis by groups like Dave Deemer at the UCSC is that Dave Deemer and Dahmer, they published a series of papers. The origins of life hypothesis is now moving away from the deep ocean to a more plausible scenario where these exact wet-dry cycles are entirely plausible at the interface of water and land. So in this connection, Jeffrey Bada, he and his group have published a couple of papers that talk about how during the late Hadean, in geological timescale, there are small islands that could have possibly been present. And those islands, which also have these hot springs that provided the path for minerals, presence of minerals, and also the interface of water and land was perfect for the wet dry cycles. And they are actually, I think he published in New Scientist, and they're also on a few field trips to hot spring areas in New Zealand, where they are actually trying to figure out if lipid, micelles on lipid membranes could be formed during at these sites of wet dry cycles, and if those things can be combined with peptide condensation, then we'll have a realistic model of how things could have evolved, things could have happened, things might have happened at the time during the times of the late Hadean. So I just wanted to add that point. If you would be interested, I would be more than happy to share those papers as well.
Yeah, thank you. Just to mention in this respect a few things. I think I forgot to mention that actually even the proteins, or at least peptides in the meteorites, were also found. So, and these are more complex proteins that were found in our experiments. So there is already some idea that origin of life chemistry has this species from the beginning. And also, I would like to mention that aminoketene molecule, if it's formed in space, it can be very simply hydrogenated. So hydrogen atoms can be added to these molecules, because you probably know most of the species in space are hydrogen. And the hydrogenation reaction is very common. And when it is hydrogenated, it became a head group of the lipid. And for me, it sounds quite exciting how basically similar species are, which have different function in our bodies, basically lipids and proteins. So if you polymerize aminoketene, it would be glycine peptide, but if you hydrogenate it, it became a lipid. And such molecules then could also act as a membrane.
Yeah, that's pretty fascinating. Um, so I have one other follow-up question. Another origin of life proposal has been like the alkaline hydrothermal vent theories, where you have relatively moderate hydrothermal vents that people have discovered, where you have a constant flux of hydrogen and ammonia from inside the Earth through small channels that usually that people have found now that possess, I think, like iron sulfur clusters, and say in a acidified ocean proposed, in the kind of pre-oxygenation event time frames. You possibly have certain types of chemistry that are catalyzed there by iron sulfur clusters, including the formation of formaldehyde from hydrogen and CO2, and potentially also amino acids otherwise. So, is there any evidence that, say, things that would coalesce in space could have catalytic functions that could help in the formation of aminoketenes or other types of similar molecules? And to what degree could you do experiments where you have catalytic, I guess you mentioned ammonia for sure, but other catalytic species that might be present at high concentrations in, say, I don't know, interstellar medium or otherwise?
Yes. Of course, yeah, the formation of aminoketene doesn't need any catalyst, because the reaction is completely barrelless and happens spontaneously without presence of any other reactants. But of course, the formation of more and more complex molecules might be influenced by the species which could be catalytic. For example, the surface of dust grains, the refractory surface of the dust grains, were proven to have a catalytic activity. For example, refractory core of the dust grains in space, it is either silicate or carbonaceous. And I think both of these materials, which when they synthesized in the laboratory with the properties as it is expected to be in space, they shown to catalyze some number of chemical reactions. So if this chemistry would take place on such surfaces, yes, they could act as a catalyst. But this requires, of course, that we don't have a very thick molecular ice mantle or covering of this refractory dust grains, because if ice would be too thick, then the influence of the surface would be minimal and could be simply neglected. But if ice is too thin or we don't have yet ice, that basically when only the first molecules are landed on the surface, then surface would act as a catalyst. Moreover, we don't know where is iron in space. So basically, it's also a riddle, which, yeah, very interesting one. But many iron-based molecule and also even iron grains were shown to act as a catalyst. So maybe iron could participate, although we don't know where is it. What else? Yeah, you mentioned sulfur. Yeah, I think some sulfur compound also for sure can contribute, but again, in the solid state, we don't know at which form the sulfur is present, and therefore it's difficult to say this for sure. But I would say there are a number of the molecules which can act as a catalyst in the interstellar medium. Yeah.
Okay, thanks.
You're welcome.
Oh, hi, Sergey. Thanks very much for sharing this very interesting research of yours. So I'm following. I mean, the previous discussion. I was just curious. Apparently you're finding a alternative route of this formation of the peptide. The is the what is the could you share like some of the stories that what inspire? I mean, because the choice of reactants. I mean, quite a unique group, and or you, of course it says abundant, but why? What leads you to focus on the aminoketene to begin with, and also the surface silicon and the potassium bromide?
Okay, thank you. Yeah, actually, it would be quite a long story to tell it complete, but I just want to say that, of course, maybe to start with, I should say that reaction of carbon atoms were almost unexplored. And we wanted to study this low temperature reaction of carbon atoms. We didn't know much yet about this, but we developed atomic carbon source, which produce those carbon atoms with low energies. And then the first experiment, we tried to mimic the condensation of this atomic carbon on the surface, on low temperature surface with other reactants. And what we found the formation of many different organic. That was first surprise because we thought that we might form either amorphous carbon, or there was some even idea that we will see some diamond grains and so on. But no, we didn't get purely carbonaceous material. We got our organic. And from this point, we started investigate the reaction of carbon atoms in more details. And why exactly carbon atoms? Just to explain, because the initial state of the matter in space is atomic gas. And this gas should condense and go to solid state, so that we have a solid material around us, finally. And this transition from the gas phase to the solid is interesting to study it. And of course, for biology and for organic, the transformation of carbon from the gas to solid is very important. So we started with atomic carbon, which is the most abundant, and then it should go to the solid state. And then came our study of the reaction of carbon atoms with ammonia, which showed us the possibility to form glycine. And then we wanted to make a next step from glycine to peptides, but it was not possible. Then we searched another possible way, and we came to the aminoketene molecules, which can be then polymerized much more simply compared to a lysine molecule.
But this is very much lacking in the previous work. How I mean, in particular, what inspired this choice?
Oh, yeah, I would not say it's so much luck. So it was an idea really to study the gas to solid transformation of carbon. So we started with carbon atoms and went to the solid state. We found the formation of organic. Then we studied specific reaction, and we were able to get the formation of glycine. And then there was, of course, quite obvious next step to go to peptides. And then, of course, yeah, that was quite maybe lucky idea that we should simply remove all water, because if we would continue like by standard pathway for the peptide formation, we would try to polymerize amino acids, and yeah, it would not be successful, but or probably it would not be successful. I don't know. But this idea with aminoketene was quite lucky. Yes, I would say. Yeah, we didn't expect that it would really polymerize at low temperature, and we just wanted to see whether we can form aminoketene at all, because it has never been observed in experiment. But besides the detection of aminoketene molecule, we were also able to see its polymerization at low temperature. So in some extent, it's of course, yeah, was a luck.
Wow, yeah, very interesting. Thanks, and congratulations for that. Yeah.
Thank you.
Yeah, thank you so much. We have been going a little bit of an hour, so I'm not sure. I know Andrew joined the stage, and Jake, if you have time for a couple more questions, and then we can close the room. Yeah, Jake, Andrew, please, and Parth, do you also want to add
Yeah,
another
thank you
comment?
again for
Yep.
doing this room. I think we are in an exciting times. I think groups like there's a group at the Scripps Institute, Dr. Ramnaran Krishnamurthy, his group, Nicholas Hudd at Georgia Tech, Lauren Williams at Georgia Tech, Dave Deemer and Bruce Dahmer, Bruce Dahmer and Dave Deemer at UCSC, and NASA Ames as a group that's working on ribosome on RNA evolution. I think we are living in exciting times, and the findings like the ones presented by this paper, I think the next 2 years we'll have more exciting results, and eventually we'll have a better understanding of what might have happened in the prebiotic world. Those are my final thoughts. But again, thank you for doing this room. I really appreciate that.
Yeah, thank you.
I also want to highlight the significance of your finding the aminoketene intermediate characterized. That is a key result because it's taken a while because they're so reactive, but to show that you've isolated them, trapped them, detected them, and they've gone on to form peptides. Wonderful result. Thank you.
Yeah, thank you. Yes.
Jake, I saw you on mic. Did you have a final question?
Can you hear me?
Yes.
Nice. I had some app issues. I had to reach in there. Yeah, I clicked in when you had spoken about clays and their ability to sort of manipulate the environment uh, to sort of guide the uh, formation of new types of things. And if I'm not mistaken, isn't that how we found the CRISPR technology with antibiotics? Didn't we take like the regular three antibiotics and stick them in sterile sand, and the next thing you know, there was like 29 other ones pop out? Or am I mistaken with that?
I'm not sure about the particular history of CRISPR, but there's a rich body of chemistry for the use of clays in catalysis and formation of. There's a number of very interesting properties that they offer chemistry, and I'm sure to speak more about it on the future. I think, particularly with respect to the subject of the origin of life, it's very fascinating stuff.
Yes, very very good room, Katerina. I really appreciate all your rooms, and I'm glad to have been able to listen to it. This was an eye opening uh, uh, meeting. I really really do appreciate you guys as wonderful minds.
Thank you so much. It's a pleasure. And yeah, this room
Can
is,
I, can I make a quick point?
yeah.
Just forgot this point. So I think to the way I see it, like many, I'm sure many think along similar lines. In the prebiotic world, there would have been multiple pathways, and we have tons of papers that are coming out in Nature Communications, eLife, and other journals. But even there, in a non-Darwinian manner, there must have been some selection, and some pathway was chosen over the others, and they were just retained. The last, eventually, when the last universal common ancestor, it's a hypothetical organism or group of organisms. We call them the Luca in the origins of life field. When the Luca eventually developed, it had all the basic recipe or ingredients of what life would have would later biology would later become not life. Life is a more philosophical entity. So, and then from the Luca, the life diverged into the three domains. That's the current understanding. So it will be interesting over the next few years, um, within all these pathways, what selection pressure would have led to eventually one outcompeting the other and then being selected to become part of biology. I just thought I'll make that point as well. Thank you very much.
I want to say thank you for sharing the fascinating work with us. Also, I mean, I just want to say one thing about the transition state because I noticed that there was two transition state on the graphs that you just shared with us on the paper. So it's another evidence for the enzymatic, I mean, kind of optimization. So this is very important, I think, because we are not talking about intermediate state, we are talking about transition state. So that was very interesting to me. Thank you for sharing.
Okay, thank you. And I would like also to thank Katerina for inviting me to present here my result. And I also would like to thank you, everybody, for sharing very interesting thoughts and ideas. And please, put all links to the articles which you were talking in the chat, so I would be able to check them. And thank you all who listen me.
Yeah, thank you so much. I'm so happy this worked out and that you came and made the time, downloaded Clubhouse. I know, but yeah, this is wonderful. And thanks for your amazing work. And yeah, enjoy the rest of your day. Everyone else, thank you for coming, asking great questions. Please come back. We will have more discussions, and I hope that Dr. Segi, you maybe also come back one day with more research updates one day, or just participate in our rooms. We have,
Yes,
yeah, and
that
that
would
would
be
be so wonderful. Um,
Could I ask
we
one more question
Yep.
while everyone's still here?
Okay.
I'm curious, along the lines of these types of tests, the origin of life and whatnot. Has there, or is anybody familiar with any types of scientific testing where they put the building blocks and added electrical charge to it, similar to how they came up with cloning?
Um I'm not sure that I correctly understood the question, but are you asking whether it is possible to try the same experiment with addition some charge, right?
Yes.
Okay. Yeah, this is actually could be quite interesting idea because we don't know how would it work. Such experiments are obviously possible, and in space we can also have some charges. And yes, indeed, such chemistry could be a bit different. But okay, we haven't tried yet. Till now, we worked with a neutral species. But yeah, maybe in the future, if we still have power to do this, yes, we can try also to do with some addition of
Oh,
charges, with cations or something like this.
that is wonderful. I'm so glad to hear about it, and I look forward to your study on whether or not it actually produces anything. That is wonderful. Thank you so very much for your time. I appreciate you.
Okay, thank you.
Okay, great. Thank you, everyone, again. And please come back, join the club. We will have a weekly recap room where we shortly just summarize what we learned throughout the week from different guest speakers. And then we will have on Monday Dr. Singh coming about his research with machine learning to how to find new rare earth compounds that we need for photovoltaic and other technology, renewable especially. Dr. Person is coming with his group. He's a physicist and how gravity emerges to present this holographic principle. And then we'll have a room about nanospacer, nano-optics, and nanospectroscopy, and a Parkinson's research room. And then Dr. Sarwood from Switzerland at IBM is coming to talk about neuromorphic computing neural networks next week. So yeah, please come back. We'll have like a roundtable discussion room later in the evening today, just discussing amongst us. And thank you so much again, Dr. Sergey. We really appreciate it. Amazing research, groundbreaking, and we are honored that we were part of history with you. Thank you.
Thank you very
Okay,
much,
thank
Doctor.
you. Thank you, everybody.
Thank you. Bye.
Bye.
I'll close the room in
Thanks,
3, 2, 1. Bye.
everyone.
Thank
Bye.
Thank
you.
you. God bless.
Thank you.